My name is Sierra Franco, and today I'm going to be discussing the KRAS protein as well as its mutation. The KRAS gene leads to the creation of the KRAS protein, which also goes by a few different names such as the CKRAS protein or the KIRAS protein. However, for the purposes of this video, I'm just going to refer to it as the KRAS protein. This protein is a part of the family of proteins called oncogenes, which have the responsibility of telling cells how to proliferate and differentiate. This means that it basically tells the cells when to grow and divide, or when to differentiate and kind of specialize in their function. The KRAS protein is able to tell cells when to grow and when to specialize because it's a relay molecule. Relay molecules are typically found in the cells traveling between the membrane and the nucleus, and they send signals from the membrane to the nucleus. That way the cell knows what to do. The sources that were provided for this project tell me that the KRAS protein and its mutation are usually found in the colon, lungs, and pancreas of humans. However, because humans and animals operate very similarly, I feel like it could be found in animals as well. There are two types of mutations that the KRAS protein can undergo, one where glycine is replaced by aspartic acid and the other where glycine is replaced by cysteine. For the purposes of this video, I'm going to be discussing the mutation where the KRAS protein replaces glycine with cysteine. In this mutation, this leads to uncontrollable growth of the cells that KRAS protein is signaling in. This is also the reason why the KRAS protein mutation is found in 10 to 30% of cancers in humans. The mutation for the KRAS protein that leads to uncontrollable growth can be found in the 12th codon. Here in the wild type, you can see the 12th codon codes for the amino acid glycine. However, in the mutation, it actually codes for cysteine. KRAS protein is a type of GTPase protein, which means that it spends its time switching from binding to GTP, then binding to GDP. When the protein is bound to a GTP molecule, that means that it's active and it's relaying signals from the membrane all the way to the nucleus of the cell. However, when it's bound to GDP, that means that all signaling is inactive and it's not really doing anything. You can see here in this model that the structure is what changes when glycine is replaced with cysteine. Knowing what we know about the protein structure as well as the mutation, we can kind of come to the conclusion that the switch from glycine to cysteine makes it either a lot harder or completely impossible for a KRAS protein to unbind from GTP and bind back to GDP and become inactive again. It can be a little difficult to see exactly where the mutation occurs with this type of model, so I also built a Lewis structure of glycine so you can see exactly what's going on. We've been over the fact that a wild-type KRAS protein will contain a glycine amino acid. This amino acid has a carboxyl group, an amine group, as well as just a lone hydrogen. This amino acid has hydrophobic as well as nonpolar properties, and is smaller than most other amino acids. This means that the wild-type KRAS protein should interact with other hydrophobic molecules, and it should also be able to form nonpolar as well as polar covalent bonds. In immune KRAS protein, our glycine amino acid would be replaced with cysteine. Cysteine also has a carboxyl and amine group like glycine does. However, it also has an extra self-hydro group. This amino acid differs from glycine not only because of its self-hydro group, but because it also is polar and hydrophilic, which would also affect the bonds and the interaction that it's able to make. Because it's polar and hydrophilic, instead of being hydrophobic and nonpolar, it's most likely going to form either polar or ionic bonds and interact with other hydrophilic molecules. Models like this 3D model of the protein, as well as the Lewis structures of both amino acids, don't really tell the full story about how proteins and their mutations actually work. These models can't effectively show how proteins interact with other proteins, whether they're wild type or mutated proteins. They also can't show the quaternary structure of the protein, which would be 3D and solid, if that makes sense. And these models also can't show how the KRAS protein would actually facilitate growth or differentiation within the cell. However, even though these models do have limitations, it's still pretty helpful to visual learners like me, so we can be able to see exactly where a protein is being mutated and exactly what it's being replaced with. For these reasons, 3D models can be pretty helpful when learning about biology.